The purposes of cloning include reproductive and research purposes. Cloning for reproductive purposes refers to the creation of a cloned organism using cloning techniques, as in the case of Kevin's dog. Other examples may include cases where a man who does not have any sperm due to a disease uses somatic cell cloning as fertility treatment, or where an extinct or endangered animal is cloned. On the other hand, cloning for research or therapeutic purposes refers to the cloning of organs for transplants or cloning of tissues to be used in research on incurable diseases. Instead of replanting the cloned embryo into the womb, removing embryonic stem cells or ES cells from the embryo and cultivating them could make it possible to grow different kinds of organs and tissues. This will be discussed in more detail in the next session. Today, I would like to focus on the ethical controversy over the application of cloning technology to humans. First, let us consider the arguments against reproductive cloning. When Dolly the cloned sheep was born, people were concerned that we would be in big trouble if someone made a clone of Adolf Hitler. However, an individual's personality is not determined just by genes. The environment in which he or she is brought up is also important. For example, monozygotic or identical twins are genetically the exact copies of each other, but they have different personalities and looks. Therefore, a clone and its original would only be identical twins whose ages differ. Even if you had your deceased child cloned, it does not mean that you could bring the same person back to life. However, there are also strong opinions that producing genetically identical individuals artificially using cloning techniques violates human dignity, and that birth by cloning is a form of asexual reproduction instead of sexual reproduction, which requires a male and female, and is contrary to the laws of nature. Another issue has to do with the low success rate and safety issues. Somatic cell cloning has a very low success rate. In Dolly's case, of the 277 cloned embryos, Dolly was the only one born. Moreover, the rates of stillbirth and birth defects, including malformation, are thought to be very high. At present, the risk may be too high for both the child to be born and the woman who would take the role of an egg donor or a surrogate mother. Secondly, in regards to cloning for research purposes, some people support its development because it could save patients with incurable diseases or patients in need of a transplant. But there are also several objections. One is that research using cloned human embryos may serve as the initial step to creating human clones, which is why it should be totally banned. Another is that cloned human embryos are also human beings, so destroying them for research is unacceptable. There is also the opinion that further experiments on animals are needed in this field of research, and it is too early to apply it to humans.